All right, so I'm very happy to introduce Athena Aktipis, who will speak to us today about cancer as somatic cheating.
Thank you so much, Anne, and thank you to all the organizers who made this conference possible, and to all of you for making the trek all the way here to enjoy some sun and also some dark rooms with slides. So today I'm going to talk about cancer in the framework of cooperation theory and conflict and cheating. And I'm going to focus in on the kinds of cheating that actually have to do with resources rather than just proliferation. The outline of my talk is I'm going to give a very, very quick review of cancer across life that sums up some of the work that we did at the Wissenschaftskolleg and the Evolution in Cancer Working Group. And that led us to sort of think about cancer in the context of what are the sort of foundations of cooperation, or the foundations of multicellularity that enable cooperation and enable multicellularity to be effective, and how do those break down in cancer? And then I'm going to talk a little bit more about resource cheating and the role it may be playing in metastasis. So we wanted to look at the incidence of cancer across multicellular life, so not just focusing on mammals or animals, but really more broadly surveying what's out there, because there has not really been any systematic study of cancer across forms of life. So our focus was on the 6 branches of complex multicellularity, but we also looked at some forms of simple multicellularity and aggregative multicellularity. To give you a sense of sort of the overall picture that emerged, in the black branches, those are the branches that are characterized by complex multicellularity. The gray are characterized by simple or aggregative multicellularity. And the red or green or yellow blocks at the end tell you if cancer was reported, which means invasion or metastasis, whether cancer-like phenomena were reported, which include differentiation and proliferation, or whether we found no reports in the literature of cancer-like phenomena. I'm going to take you very quickly through some of the examples here. So in bilateralia, we observed cancer in lots of different species, or we found reports of cancer in lots of different species. In Cnidaria, we saw a loss of differentiation in coral and actual full-on cancer in Hydra. In fungi, we saw cancer-like phenomena in both branches of complex multicellularity that are fungal. In green algae, including plants, we see both abnormal proliferation and cancer-like phenomena, as well as there have been reports actually of metastasis or metastatic strands in plants as well. So that was actually surprising because there was a recent review, I think in Nature Reviews Cancer, saying that it's not possible to have cancer in plants, but there are some early reports in the literature of cancer in plants. In red algae, you see cancer-like phenomena. Brown algae, similarly, cancer-like phenomena. And we looked at some branches of simple multicellularity, and in many of them we didn't see cancer-like phenomena, and some we found some cancer-like phenomena, no full-on invasive metastatic cancer. We're also looked at a few examples of aggregative multicellularity. So these are organisms that can be either unicellular or multicellular, depending on the life stage. And we see reproductive cheating and resource monopolization, which actually sort of examining these aggregative multicellularity these species that are characterized by aggregative multicellularity help to kind of guide us along in terms of thinking of some of these bigger issues with how resource use may be related to cancer more broadly. So again, here is the sort of overall pattern. This paper is right now under review at Philosophical Transactions B to appear in a special issue on Peto's paradox and comparative oncology. So I'll post that on my website if and when it becomes available. So to kind of back up a little bit to the big picture here, is cancer cheating really in the cooperation that enables complex multicellularity? So we could think about multicellularity as a few different forms of cooperation all happening together. And the more complex the multicellular body, presumably the more you have to do of all of these things. And what happens when cheating occurs in these sort of foundations of multicellularity? Well, in fact, the phenotypes of cancer actually fit very well with a breakdown of these cooperative phenotypes. So we decided to sort of pull this all together into a framework, which we call the sort of foundations of multicellularity. And I need to give credit here to Amy Body because she's the one who made all these amazing figures that I'm about to show you. So so we see that there are 5 main components of multicellular cooperation that really enable the multicellular bodies to get more and more complex and large. This includes inhibiting proliferation, controlling cell death, but also more sort of economic forms of cheating like, economic forms of cooperation like allocating resources and transporting resources, dividing labor, and creating and maintaining an extracellular environment. And so when those break down, we see cancer phenotypes. And with proliferation cheating, there's a number of aspects of cancer that fit with that. I'm not going to go through and read all these, but they include generation of growth signals, avoiding senescence and apoptosis, et cetera. I'm sorry, senescence and differentiation. So cell death cheating, this is basically any avoidance of the cooperative multicellular mechanisms that regulate cell death in order to benefit the whole multicellular organism. Then you have resource cheating, and we know that in order to build a multicellular body that is larger than really just a few hundred cells, you need some form of resource transport. Otherwise, the cells on the inside won't have resources because they can't get them through diffusion. And so this resource transport and sharing is actually a very important component of multicellularity. And in cancer, we see a breakdown of resource transport and resource sharing and monopolization of resources by cancer by angiogenic signaling and alterations in metabolism that allow the cancer cells to take up more resources than their less neoplastic neighbors. Labor performance and division. This is another really important aspect of multicellularity, not just sort of division of labor reproductively, but also division of labor into many different cell types to create different organs to allow for much more complex function. And then the extracellular environment is, of course, very important. We're made up of cells, but we're also made up of a lot of things that our cells, excrete in order to make our bodies function. And normal cells help to create that environment and help to maintain it. Cancer cells do the opposite. They break it down, they use it for their own growth and proliferation, and they break it down also traveling through it to invade and metastasize new organs. So we've sort of placed these aspects of somatic cheating in 2 categories. So the first one is really what we are calling demographic cheating, which is related to cell proliferation and cell death. But then we also have these more sort of economic forms of cheating that have to do with resources, labor performance, and the maintenance of the shared environment. Now, how does this framework fit in with sort of already accepted frameworks for cancer? So there's a very famous framework for cancer called the hallmarks of cancer. And what we decided to do was see how these aspects of multicellular cooperation and cheating fit in with the hallmarks framework. And so what you're seeing on the outside here is the hallmarks framework, and on the inside, the foundations of multicellularity framework. And turns out that 3 of the hallmarks of cancer relate to uncontrolled proliferation. 2 of them relate to inappropriate cell survival. 2 of them relate to resource monopolization. And one of them relates to the sort of environment issue, which is the invasion side, although metastasis is included in this. And I'm going to talk about metastasis in a moment. I think that in order to understand metastasis, we actually have to go a little bit further than this. I also want to point out that on the very bottom, we think this might indicate a missing hallmark. So the breakdown in division of labor really is manifested by dysregulated differentiation. And we saw that in a lot of the sort of cancer-like phenomena across life that we looked at. But dysregulated differentiation is at least not currently one of the hallmarks of cancer, but perhaps it should be. So one of the aims of my work is really to look at whether economic cheating is related to invasion and metastasis. And I'm going to briefly show you the results of a model here where we found that as rate of metabolism went up, so as the cells consumed resources more quickly, their motility rate went up. And this was the case for neoplastic cells that had higher metabolic rates, but not for normal cells in this agent-based model. We also did a sort of similar model where cells were able to move in response to resource limitation, and so they had a sort of threshold for moving. And what this meant was that the threshold, we allowed the threshold for movement to evolve. And what we found is that the higher the metabolic rate of the cells, the higher that threshold was, which meant that the cells were more likely to move even if the environment was not that empty of resources. So they're just more ready to move in general. So what we wanted to sort of do then is to take these ideas and again see, can we pull them together with some cooperation theory. So some of you might know my work on walk away, which is this very simple rule which says stay if you're in a cooperative environment, leave if you're in an environment with defectors. It's basically a social generalization of the simplest foraging rule. So stay if things are good, leave if things aren't so good. And so we've actually wondered whether in order to understand what happens in advanced cancer, perhaps there is actually selection that might favor sort of construction of new niches by cancer cells that favor the cells that are their sort of their clone. And that might partially be due to walk-away-like dynamics. So you can sort of imagine in the sort of unfavorable environment, so you can imagine early in cancer progression, resources are being depleted, there's waste products, unrestrained metabolism, the environment is not so good, so you're likely to get movement. Because cells are actually able to respond, even non-cancerous cells are able to respond to resource levels in their environment. So you can get upregulated movement there. Now, in a more favorable environment, you're going to have a sort of more stable and long-lasting set of cells. And that might be early progression, but that might also be late in progression if the cancer cells can sort of re-evolve to cooperate, but only with themselves, in a sense. And this sort of leads us to think even more broadly then about how does selection act on metastatic capacity. So, it could very well be that initially you have a primary tumor and you mostly have resource competition dynamics going on. Perhaps you can get actually selection for multiple generations of, or you can get multiple generations of metastases where each of those generations is actually getting selected for more cooperative within micromet features. So maintenance of a shared environment, maybe you even get some restrained proliferation in order to sort of benefit the little microcolony of tumor cells. So this is a model that we, sort of verbal model, that we're publishing now in a chapter on Frontiers in Cancer Research, and it's with Josh Schiffman, who's also here. So we wonder whether really kind of taking things all the way through from, initially multicellularity. In order to evolve, you need cooperation. That can break down with initial evolution of cancer, but then perhaps within those metastases, or within those neoplasms, within that process, there may be selection for cooperation again that could be important in understanding that process. Thank you.